Hello students, myself Dr. Sonawane, and today we are discussing how to estimate the heritability by half-shift correlation methods. When we are talk about the heritability, this is very important genetic parameters to study the genetic variation present into the population. This parameter heritability was originally given by Dr. G.S.Sonawane along with heritability and genetic correlations. When we are discussing heritability, there are two different types. One is broad sense heritability and second one is narrow sense heritability. And both notations, capital S square and small s squares, they are taken from the Swell-Wright dictionary. When we are talking about the broad sense heritability, it is genetic variance to phenotypic variance, and it includes genetic variance due to the additive genetic variance, dominance, as well as epistasis. But when we are talking about the narrow sense heritability, it is always additive genetic variance only with phenotypic variance. So when in general we are refers to the heritability, the default meaning is narrow sense heritability. Here we are discussing about the range of heritability between 0 to 1. When low heritability, it starts from 0 to 0.15, and under that, trait related to the fitness like survivability and reproductive traits are coming under that. Then medium heritability having the range 0.15 to 13. And if we see that the production traits mostly coming into the medium heritability traits like milk production, egg production, wool production, etc. Then in case of high heritability traits, 0.3 to 0.7 and above, under that the showness traits are coming or qualitative traits, example carcass traits or color, etc. They are coming under that category. When we are talking about different methods to estimate heritability, there are two important methods. One is regression methods, under that subtypes like regression of offsprings on one parents, regression of offspring on the mid parents, and intrasire regression of daughter on dam. And in second methods, correlation methods, there are two important half-shift correlation and full-shift correlation methods. When we are talking about the full-shift and half-shift, in full-shift correlation methods, there are two important aspects, C effects and maternal effects. And because of C effects, that is common environmental effects, they have more similarity. And again, they are showing the less variation, or we can say that the efficiency to find out heritability is somewhat less in case of full-shift as compared to half-shift. Then again, because of maternal effects, pre and post, particularly litter bearing animals, they have much more similarity, and these two factors hampering the heritability. That's why we geneticians most preferably go for half-shift correlations. In case of half-shift correlation, there are two important aspects. One is paternal half-shifts and another one is maternal half-shifts. In case of paternal half-shifts, we are generally used because different sires are used in different farms, and through their progenies performance or their records, we are calculating the heritability for that concerned sires. Then next, when we are talk about the paternal half-shifts, here each number of sires, they are mated with a large number of dams allotted at random, means there is no choice, but we are selecting the dams from different herds or same herds randomly. And one offspring is produced from each dam, and all such offsprings from one sire are known as paternal half-shifts, where one sire is combined for all. These half-shifts have a coefficient of relationship 25% on an average because they receive the 25% genes common from the common sires. With the help of analysis of variance, we can find out the value of sire component for that variance and to get the exact value of heritability estimate. The next, what are the advantages of paternal half seeds? Most important, it does not indulge in the dominance effect in variance. It can be used for the characteristic. They are sex limited or carcass traits which getting after the destruction of animals. Then performance records of all daughters are at same period. So actually, automatically, the uniformity in environment related to the time and management we will get, and influence of mating systems can be excluded by these methods. Then what are the limitations? Sire component of variance is to be multiplied by 4 to reach the expected value of heritability, and it will inflate or reduce the value of heritability to great extent. Multiplication by 4 also increases the value of standard error, and again when there is a number of sires are less and their offsprings are also less per sire, then it will reduces the efficacy of method or increases the standard error. This is the ANOVA table for estimation of heritability. In first column, source of variation, under that between sires, within sires, as well as totals. Then degrees of freedom. Here we are minusing 1 from the total number of sires. In case of within sires, total number of records of the progeny is -1, minus number of sires to get within sires degree of freedom, and in case of total number of observations for progeny is -1. In case of sum of squares, we are getting sum of squares between, sum of squares within, and sum of squares for total. Then mean sum of squares is calculated between sires by sum of squares between sires divided by S minus 1, that is degree of freedom for between sires. In case of mean sum of squares within divided by degree of freedom for within sires. The next point is expected mean squares, EMS. Here variance between plus variance within are added, and in case of within sires only within variance between sires is calculated. For final heritability estimations, we are multiplying by 4 to the variance between sires divided by variance between and within sires. Here, small n is related to the number of sires and capital N is related to the total number of records for the progenies for each sires. Then component of variance within the sire component of variance is equal to mean sum of squares within. In case of between the sire component of variance, between variance is equal to mean sum of square between minus mean sum of square within divided by k. Here k is average number of progenies per animal. For unequal number of progenies per sire, k can be calculated like that. K is equal to 1 divided by S minus 1, where S is related to total number of sires, capital N is related to the total number of progenies, and Ni related to the number of progenies per sire. Now we will take one example, through that we will get idea how to calculate heritability by paternal half seeds. In this example, we are estimate the heritability for first year old yield, and the value are given in kilograms by paternal half seed of 4 Deccan rams. So ram number 1, 2, 3, 4, and each have 10 progenies, and their respective first year old yields is given in this example. When we are seeing, this is the basic calculation table for RAM 1 to RAM 4 with their 10 progenies. First, we add all these observations. So for RAM 1, total number of progenies 10, RAM 2, RAM 3, RAM 4 also 10. Then we are making a summation of all observations for RAM 1 from all their 10 progenies, same way for 2, 3, and 4th RAM. So we are getting these values. Then again, we are going to summation of square of each value and summation of all these for each RAM. And then whatever the values here, we are again squaring to get these values. Then coming towards for making the value in analysis of variance, first we are finding the correction factor by whatever values we are getting in these tables. We are squaring for each values by summing and dividing by total number of observations. So we are getting the correction factors here 18.15. The next, total sum of squares. Here again, crude sum of squares minus correction factors. How to calculate crude sum of squares? By summation of all values for each column under each side or RAM, and then adding and minusing the correction factors. So we are getting the value of total sum of square 0.80. Then between component of sum of squares is again doing by squaring summation of all values divided by total number of progenies for each side minusing correction factors. We are getting value 0.11. Then next is within component of sum of squares. Here again we are minusing sum of squares between sides from sum of squares due to the totals, and we are getting sum of squares within 0.69. Then next we are filling the ANOVA table with these values, and we go to calculate the variance between sides and within sides. Then again, component of variance within the side or within the RAM components, we are getting 0.019, and between the side component of variance, we are using that formula, mean sum of square between minus mean sum of square within RAMs divided by K. Here K are equal number of progenies, that's why 10 we are taking, and we are getting the value of variance between the sides is 0.0018. Then we are put these values into the heritability estimation formula by multiplying with 4, and we are getting heritability 0.348. So this is the heritability for full production of first year for RAM. So I think you are getting how to calculate the heritability by pattern and half shifts. So in this way, we are calculating the heritability by pattern and half shifts. Thank you.